Hello, my name is Kaylee Knutson.
And I'm Sarah Al Nasrallah.
Today, the topic we will be talking about is meiosis. So, the purpose of meiosis is to create sex cells called gametes. This occurs in sexually reproducing eukaryote organisms, and the reason why meiosis is so important to biology is because meiosis creates genetic diversity, which I will talk about further, in eukaryotic organisms, providing a stronger offspring, allowing it a chance of surviving in the world. Meiosis is also important because this process also answers how we obtain genetic diversity and why we don't look exactly like our parents. So, genetic diversity is basically diversity in organisms, and they're not going to look exactly like their parents because they are going to obtain different kind of genes and everything in every single offspring that the initial parent has. So the reason why gametes are produced by meiosis is because meiosis allows for genetic diversity. Each daughter cell is different from each other and the parent cell. This is because of independent assortment in metaphase 1 and crossing over in prophase 1. There's an error. I will fix that. And I will go into independent assortment and prophase 1. And crossing over in further. Okay, so how does meiosis occur? Meiosis occurs in sexually reproductive organs such as the testicles and ovaries. The cell that will divide by meiosis are called diploid germ cells. These germ cells will then go through a two-part cell division, which will then create gamete cells. Gamete cells are haploid cells. So the terms that will help you further understand this topic are haploid cells, which that is a cell that has only one set of chromosomes. And a diploid cell is a cell that has, two sets of chromosomes. A homologous pair of chromosomes are a pair of chromosomes that share the same sequence of genes for traits, and they look alike, essentially. Alleles is a part of a homologous pair that might have different alleles, which alleles are basically variations of the same gene. And nucleus is the center of the cell. In eukaryotes, the nucleus stores DNA and facilitates replication and transcription. Germ cells are diploid cells that divide during meiosis, and gamete cells are basically our end product of meiosis. And they are haploid cells, so they're only going to have one set of chromosomes. And another name for, gametes are sperm and egg. These, as I said before, they're created by meiosis. Crossing over is basically a homologous pair of chromosomes exchange genetic material, and this happens during prophase 1. Independent assortment is chromosomes will arrange themselves randomly during metaphase 1. So now we will finally get into the steps of meiosis. And there is actually 2 stages of meiosis, right? And they all have different phases. So first, in the first stage of meiosis, which is titled meiosis 1, there's prophase, and the chromosomes condense, the nuclear envelope breaks down, and crossing over will occur. Metaphase 1, which is the next step, pairs of homologous chromosomes move to the equator of the cell, and then there's anaphase 1, and the homologous chromosomes move to the opposite side. Oh, and also in metaphase 1, the chromosomes will align in the equator of the cell, but they'll also stack on top. As you can see, the red and the blue, those are two separate sets of chromosomes, and they're stacking on top. And so the homologous pair of chromosomes in anaphase 1 are going to move to opposite sides of the pole, and they will be being pulled by spindles that are released from centronomes. And then the last phase before meiosis 2 occurs is telophase in cytokinesis. And that is basically, telophase is the cell, the nuclear envelope is now redeveloping, and the cell, and cytokinesis is basically the cell finally like pinching off. And there's that cleavage right there. And so meiosis 2 is basically the same thing, except you have two cells, if you started off with one, and in metaphase 2, the chromosomes will stack alongside each other, and they're not going to do this, stacking on top as well as stacking alongside. And they are going to be ripped apart, divided in anaphase, and then telophase 2 and cytokinesis. There is going to be, the nuclear envelope is being created again, and the cell is now dividing, right? So then you're going to end up with 4 cells. So basically, interphase, which I should have said, happens even before meiosis can occur. Now, interphase is basically when the DNA is being synthesized, which you need that, and the cell is literally growing. And so there are 3 phases. There's G1, which is the first phase, and that is when the cell is going to increase in size and prepare for division. In the S phase, which you'll notice that the S stands for synthesis, and DNA is beginning to, to synthesize. And in G2, that is the last stage before meiosis, and the cell is going to be, continuing normal function and continuing to increase in size before it even goes into meiosis. All right, now we're going to go deeper into what prophase exactly is. So in prophase 1, the chromosomes, they'll start to condense, and the nuclear envelope will start to break, as you can see from the little brown chunks that are circling the red and blue. Anyway, so as the nuclear envelope starts to break, the centrosomes, right, they will start to release spindles, which are the gold little lines, and they're going to start moving to the opposite side of each cell so they can prepare to latch on to the middle over the middle of the chromosome. And this is also when crossing over will occur. And crossing over is when, right here, crossing over is the cellular process that happens during prophase 1, when chromosomes of the same type are lined up. When 2 chromosomes, one from the mother and one from the father, line up, parts of the chromosomes can be switched. So like different alleles can be switched and everything. The 2 chromosomes contain the same gene, but many have different forms of genes. Right. Okay. So as crossing over occurs, and the centrioles will start to migrate to the, okay. All right. So that is prophase 1. Now the difference between prophase 1 and prophase 2 is basically crossing over does not happen, but the same sequence of events happen. Okay, covered crossing over. So metaphase is basically the second phase of meiosis, right after prophase. And during metaphase, the chromosomes will line up and prepare for separation. The centrioles will let out spindles and attach themselves to the centrosomes of the homologous chromosomes, as you can see right here, and they are stacking on top of each other. So in metaphase 1, the chromosomes will stack on top of each other and, go to the equator of the cell. During this, independent assortment occurs. And independent assortment is the process where chromosomes move randomly to the separate poles, and during metaphase 1, as you can see here. And the difference between metaphase 1 and metaphase 2 is that the chromosomes don't stack on top of each other in metaphase 2, and also independent assortment does not happen in metaphase 2. Only in metaphase 1. And now my lovely partner will take it from me.
For anaphase, the sister chromatids will separate at the centrometers, and the daughter chromosomes move to the opposite poles of the cells. The cell also elongates as the chromosomes detach from one another. In anaphase 1, the chromatids are still intact. It's an X shape, whereas in anaphase 2, when they're detached from one another, they form an S shape rather than X shape. And in telophase and cytokinesis, in telophase, the cell continues to elongate. The nuclear envelope forms around the chromosomes. The chromatin unfolds and the nucleoid reappears. And then in the cytokinesis stage, the cytoplasm is divided between the two daughter cells, and the cleavage forms to separate the two cells. And then in cytokinesis 1, we'll end up with 2 daughter cells, whereas in cytokinesis 2, we'll end up with 4 daughter cells. How we could relate this to the world is with genetic disorders that can come if meiosis doesn't end up happening properly. One of the genetic disorders are XXX. It's a syndrome where people can have 3 X chromosomes. It is only in the females, is very rare, and it can come with a lot of symptoms. And for example, like physically, you would have weak muscle tones, could have seizures, problems with kidneys, and premature failure in the ovaries. So people would not be able to have children, and they'd be infertile. And then psychological problems would be delayed development. They could have dyslexia and ADHD, depression, and anxiety, or they could have all of them. This can be very costly for the parents since they would have to pay for treatments, whether it be problems with the physical self or the mental state of their children. We chose this topic because it was something that was hard for us to grasp when we first taught it, especially for me. I got a few questions wrong on the test, and I thought it'd be best to focus on this so we would be able to reach a deeper understanding. And we did a problem explaining meiosis, and we linked the video in this slide. And that's it. Thank